Welcome to Biochemistry of Metabolic Processes. Today, we are diving into glycogenesis, the fascinating biological process our bodies use to store energy for the future. Imagine eating a massive meal. Your body needs a way to save that fuel for later, rather than letting it go to waste or cause harm. We will explore exactly how your cells act like microscopic bank vaults, packing away sugar molecules safely and efficiently. Let us get started. To begin our journey, let us think about how we handle money. Imagine you just received your monthly salary in cash. If you keep all those loose bills in your pockets, your wallet will become dangerously overstuffed, and you might even lose some of it. Instead, you take the bulk of that cash and deposit it into a secure bank vault for future use. In the human body, the sugar called glucose is exactly like that loose cash, providing immediate energy for your cells. When you eat a large meal, your blood becomes flooded with glucose. Leaving it all in the bloodstream is dangerous, so your liver and muscle cells act as biological bank vaults, packing the individual glucose molecules together into dense chains called glycogen. Now that we understand the analogy, let us look at the formal biological definition. Glycogenesis is the specific anabolic metabolic pathway through which individual molecules of glucose are chemically linked together to form glycogen, which is a complex carbohydrate polymer. This vital process takes place primarily in the cytoplasm of your liver and skeletal muscle cells during periods when blood glucose levels are elevated. The pathway is heavily regulated and is primarily stimulated by the hormone insulin, which signals the body that energy is abundant and should be stored. Biochemically, the process does not just stick glucose molecules together directly. It requires a series of enzymatic steps to first activate the glucose using cellular energy before it can be stored. Understanding what glycogenesis is naturally leads us to ask why it is so incredibly important for our survival. If our bodies lacked the ability to perform glycogenesis, our blood sugar would spike to toxic levels immediately after eating and then crash to lethal lows just a few hours later. Furthermore, by converting highly soluble free glucose into large, insoluble granules of glycogen, the cell prevents excessive water from rushing in via osmosis. This ultimately protects the cell from swelling and bursting while safely hoarding fuel. In skeletal muscles, this stored glycogen serves as a private, on-demand energy reserve that allows for sudden, intense physical activity, like sprinting away from a predator. Without it, humans would have to eat constantly. This biological necessity brings us to the actual chemistry of the storage process. Here is the overall chemical equation for adding a single molecule of glucose to a growing glycogen chain. Conceptually, this formula shows us that storing energy is not free. It requires an initial investment. Let us break down the symbols. G represents a single molecule of free glucose entering the pathway. ATP and UTP are the high-energy currency molecules of the cell. Gly-N represents the existing glycogen chain before the new glucose is added, while Gly-N+1 represents the newly lengthened chain. Finally, Pi represents inorganic phosphate, which is released when the energy-rich bonds are broken. If cellular ATP decreases, this pathway slows down as the cell prioritizes immediate survival over storage. Building on that chemical equation, we can derive a simple formula to calculate the total metabolic tax the cell must pay to build its energy reserve. Conceptually, Because the activating enzymes use one ATP and one UTP respectively, and UTP is energetically equivalent to ATP, there is a strict cost of 2 ATP equivalents per glucose molecule. In this formula, CT represents the total energy cost measured in ATP equivalents. The variable N represents the total number of glucose molecules that need to be stored. The number 2 represents the constant biological cost of 2 high-energy phosphate bonds required to process each individual glucose molecule. If the number of glucose molecules increases, the total energy cost increases proportionally. Before we move on, let us see this formula in action with a concrete example. Suppose a skeletal muscle cell has just taken up 150 molecules of free glucose from the bloodstream after you ate a large bowl of pasta. The cell needs to store all of these molecules as glycogen to prepare for a heavy workout. We are asked to find the total high-energy phosphate bonds required. We choose our total cost formula because it directly relates the number of molecules to the ATP equivalents needed. We substitute 150 for N and multiply by the constant cost of 2. This gives us a final answer of 300 ATP equivalents. Physically, this means the muscle cell must spend 300 high-energy bonds just to pack the glucose away. Now that we have calculated the energy cost, we must address a critical thermodynamic challenge in this pathway. During the activation step of glycogenesis, a modified glucose molecule reacts with UTP to form an activated intermediate and a molecule of pyrophosphate. The problem is that the standard free energy change for this specific reaction is approximately 0. In biochemistry, when a reaction has a free energy change near 0, it sits at equilibrium, meaning it goes back and forth equally. It is fully reversible. If the reaction can go backward, the cell might fail to build glycogen when it desperately needs to. The cell must find a way to force this reaction to move strictly forward to ensure glycogen is successfully built. To solve this equilibrium problem, the cell employs a brilliant thermodynamic trick called reaction coupling, which brings us to this formula. Conceptually, the cell uses a special enzyme to immediately grab the pyrophosphate product and chop it in half. This secondary cleavage reaction releases a massive amount of energy, acting like a vacuum that pulls the first reaction forward. In our formula, delta G OVR represents the total overall energy change of the combined two-step process, measured in kilojoules per mole. Delta G act is the energy change of the initial activation step. Delta G CLV represents the energy released by breaking apart the pyrophosphate molecule. If the cleavage energy is highly negative, the overall energy change becomes highly negative, driving the process forward. Let us apply this coupling formula to see exactly how much thermodynamic pull is generated. We are given that the energy change of the initial activation step is roughly 0 kilojoules per mole. We are also given that breaking apart the pyrophosphate molecule releases a highly negative energy of negative 19.2 kilojoules per mole. We need to find the overall free energy change. We use our reaction coupling formula because it combines these two sequential steps. By substituting 0 for the activation step and negative 19.2 for the cleavage step, we simply add them together. The final answer is negative 19.2 kilojoules per mole. Physically, this highly negative value means the combined reaction releases a lot of energy into the environment. So, we've calculated the overall free energy change. This brings us to proving exactly why this guarantees the pathway only moves in the direction of storage. In this first part of the proof, we set up the chemical logic. We start by defining the activation reaction, which produces UDP glucose and pyrophosphate. Because its free energy change is zero, the mathematical equilibrium constant allows the reaction to easily reverse. To prevent this, we introduce the secondary cleavage reaction, where pyrophosphate reacts with water to form two inorganic phosphates. We note that this specific step has a highly negative free energy change of negative 19.2 kilojoules per mole. This setup is crucial because it establishes the two components we need to combine in the next step. Continuing our proof, we now combine the two reactions to see the final result. Mathematically, because pyrophosphate is produced in the first step and immediately consumed in the second step, the pyrophosphate term cancels out from both sides of our combined equation. This leaves us with a net reaction that directly produces UDP glucose and two inorganic phosphates. Because we proved in our example that the overall free energy change is negative 19.2 kilojoules per mole, which is strictly less than zero, the laws of thermodynamics dictate that this net reaction is strictly spontaneous. The intuition here is that by intentionally destroying the pyrophosphate byproduct, the cell permanently removes it from the environment, making it impossible for the activation step to reverse itself. Now that we have proven why the pathway only moves forward, let us look at the complete biochemical pathway of glycogenesis. At the very top of the diagram, we start with a single molecule of free glucose entering the cell. Following the downward arrows, you can see the initial investment of ATP, converting it into glucose 6-phosphate, which then rearranges into glucose 1-phosphate. Next, notice the critical activation step we just discussed. UTP is invested, and pyrophosphate is released and immediately split into two inorganic phosphates, driving the formation of UDP glucose. Finally, at the bottom, enzymes called glycogen synthase and branching enzyme take this activated UDP glucose and attach it to the growing, highly branched glycogen primer. This diagram beautifully summarizes the entire elegant process of energy storage. We have covered the full topic of glycogenesis, so let us reinforce the most important ideas. First, glycogenesis is the essential metabolic pathway that safely stores excess glucose as glycogen, acting as a crucial buffer for human energy homeostasis. Second, we learned that this storage is not free. It requires a strict investment of 2 ATP equivalents for every single molecule of glucose packed away. Third, we saw how the cell uses brilliant thermodynamic reaction coupling, specifically the cleavage of pyrophosphate, to make the entire pathway strictly irreversible. By mastering these concepts, you now understand how our bodies protect cells from osmotic stress while guaranteeing that our brain and muscles always have a reliable supply of fuel. Excellent work today.